This method can help answer key questions about integral membrane proteins, such as the oligomeric state, their size, overall shape, and low-resolution structure in solution. Solution studies of membrane proteins require stabilizing molecules like detergents. This technique has the advantage that it makes detergents invisible and highlights the signal from the membrane protein.
The implications of this technique extend to many biological and medical research questions because over 1/3 of proteins reside in membranes, where they perform many vital cellular functions.
Visual demonstration of this method is critical as the deuterium labeling and neutron scattering steps are difficult to learn because very few institutions offer training in these techniques.
To begin, determine neutron scattering length densities, or SLDs, using the web application Mulch modules for the analysis of contrast variation data. Open the Contrast module by clicking Contrast, located in the left navigation pane. Provide text for a project title and enter the details below for two components as Subunit 1 and Subunit 2. Enter the molecular formula for each component in the formula text box. Then, enter the volume in cubic angstroms for each component in the box below volume. Finally, enter the details for buffer components. Click Submit to perform the neutron contrast calculations. A results page is generated that provides a table of scattering length density and contrast match points, as well as formulae for determining these parameters at any given percentage of D2O in the buffer. Review the scattering parameter.